Last class, we have completed Entamoeba histolytica and Trypanosoma gambians. In this today's class, we are going to finish this malaria and we're going to solve some of the questions with respect to the protozoa. Clear? Okay. We have completed up to this. Up to this slide, we have completed yesterday. From now, this slide, liver isogony. See, when it comes to malaria, you have to remember these two important pictures. This from straight away they can ask the question from the this picture. What are all the things you have to remember is the two most important for malaria is for this protozoan diseases is man, human as well as the mosquito. Clear? Okay. In this mosquito, what are all the target areas they have been targeting? Gut and the salivary glands. Okay, this is the second thing you have to remember. After remembering the host as mosquito and human, what is the other thing you have to remember? In mosquito, the targeted area are gut and the salivary glands. When it comes to human, the targeted areas of this parasite is liver and the bloodstream. Okay, now after remembering these things, first thing is host and the second thing is targeted areas in that host, and the third important thing is what is the cycle takes place by the parasite in man as well as the mosquito. Okay, in man, asexual cycle. In mosquito, it is a sexual cycle. Okay, this is the third thing you have to remember. And the fourth thing you have to remember is what is the end product of each target area. In liver, the end product is merocyte. In blood, the end product is gametocyte. In mosquito, the end product is sporocyte, and this sporocyte is the thing enter into the salivary glands of the mosquito. And these are the thing you have to remember when it comes to the end product. And the fourth thing you have to remember, keep in mind is the end product of each targeted area. And the fifth important point you have to remember is which is the important infectious agent among these end product, and which is the infective agent, and that is nothing but the sporozoite. And that infective agent, the infective stage of the parasite is called as sporozoite, and that sporozoite will reach the salivary glands when it bites when the infected mosquito bites the normal healthy person. This sporozoite will enter into the liver cell of the human, and that sporozoite will convert it into the merocyte in the liver, and that merocyte, which is in haploid in nature, they enter into the bloodstream, and now there the merocyte will be converted into the tropocyte, and again the tropocyte will be converted into the merocyte. Here one of the cycle will continues. Okay, by the process of zygogony. That zygogony is nothing but the asexual reproduction mechanism, and that too multiple fission. The zygogony is the type of the multiple fission. You know the basic difference between the binary fission and the multiple fission. And now this merocyte is develops into the gametocyte, male and the female gametocyte. Very good. Male or the female gametocyte. Now this gametocyte is the thing when the mosquito bites an infected person. Now this gametocyte will be taken up by the mosquito, and that gametocyte will reach the gut of the mosquito. Now that female and male mosquito, again they are going to undergo a reproduction mechanism, that is fertilization. Male and female fuse together to form a 2N. See, everywhere we have seen only haploid cell. Only after fertilization they are going to form the zygote. In zygote they are always in the diploid nature because of the fusion of the one male and one female gametocyte. Clear? Now, in that gametocyte, they are once again they are going to convert into the ookinete or the oocyst. Okay, I'm going to explain what is ookinete or the oocyst. Now, this oocyst is the one again converted into the sporocyte. Now this sporocyte is enter into the salivary glands. Now it is ready to cause the infection, and it is ready to complete its asexual reproduction when it reaches the human. Okay, this is about the simple explanation of the malarial parasite. Clear? Fine. Again, the same thing has been repeated, little more elaborate here. Now, coming back to today's session, liver cysogony, the process which is going to see takes place, the what are all the process takes place within the liver, it is called as the liver cysogony, and what are all the process which is takes place in the blood, that is said to be as the erythrocytic cysogony. Why we called as liver cysogony and erythrocytogony means the process, the asexual reproduction mechanism followed in this type of malarial parasite is cysogony. That is why the reproduction takes place in the liver. It is called as the liver cysogony, and the reproduction, asexual reproduction, which is takes place in the blood, it is called as the erythrocytic cysogony. And cysogony is nothing but a type of a multiple fission. Clear? Okay. Now, liver cysogony. Once within the human blood, the sporocytes circulate for about half an hour. Okay, and then get into the liver. Once it is enter, once it is get, once a normal healthy person got bit by the mosquito, the parasite will enter into the human blood and it circulate for about half an hour. And what is that infective agent taken by the normal healthy person from the infected person is from the infected mosquito is sporocytes, and that will circulate about half an hour, and only after half an hour they'll reach the liver. To reach liver it takes about half an hour, and after reaching the liver it reaches the hepatic cells. We know liver is made up of a liver cells, and liver cells is called as the hepatic cells, and they are going to multiply. And what is the mechanism? Asexual reproduction. And what is the type of asexual reproduction seen in this reproduction mechanism is cysogony, and cysogony is nothing but the type of the multiple fission. Clear? Fine. Now in this liver cysogony, there are two different phases can be seen. Pre-erythrocytic phase and exoerythrocytic phase. Pre-erythrocytic phase means before it enter into the blood. Okay, and exoerythrocytic phase means now the phase, now the what are all the end product synthesized in the liver is ready to enter into the RBC. That is called as exoerythrocytic phase. Now the what are all the things which has happened in this phase called pre-erythrocytic phase means after now the sporocyte will reach the hepatic cell. Now this each sporocyte will becomes a cryptozoite. It undergoes a structural changes. Okay, and how this cryptocyte will look like means it grows for a number of days and becomes spherical, non-pigmented cyzone. That is said to be as the cryptozoite. See, sporocyte is an infectious agent. Now it is ready. Now it is reaching the liver cell. In their liver cell, they are definitely they are going to undergo their multiplication, their reproduction. In that reproduction, what are all the structural changes they are undergoing? Means this sporocyte will become the cryptocyte, and the cryptocyte will look like the spherical non-pigmented cyzone. Okay, now after converting into the cryptocyte from the sporocyte, this cryptocyte is divides by the cysogony, multiple fission, and forms a large number of uninucleate cells called the cryptomerozoites. See, initially it was sporocyte. This is called as the infectious agent. After reaching the liver cell, it is converted into the cryptozoite. And after the cryptozoite has been formed, this cryptozoite is now ready to undergo the asexual reproduction called the cysogony. And this under the process called the cysogony, this cryptozoite is divided into the multiple cells which possess the single nucleus, and that is called as the cryptomerozoites. Okay, now they are liberated when the liver cell bursts. The liver cell has the capacity to hold limited number of cells, but under the process of physogony, they are giving rise to many types of cryptomerozoites. Now, the liver cell can't have the capacity to hold every cells. So now the liver cell is going to burst. When the liver cell bursts, every cryptomerozoites will be liberated. Now, in this phase, the blood remains sterile and its inoculation does not produce infection. See, the cryptomerozoites does not produce any type of infection. That is why we are saying the sporocyte is the only thing going to cause the infection. Clear? The blood remains sterile and its inoculation does not produce the infection. Now, in the exoerythrocytic phase, the cryptomerocytes enter the fresh liver cells. After completing the one liver cell, it enters into the another liver cell. There, they are going to convert this cryptomerocyte into the metacryptozoids. Okay, see, initially it was a sporocyte. Next, they are going to convert into the cryptomerocytes. Again, they undergo the process of asexual reproduction called zygogony, and they are converted into the cryptomerocytes. And this cryptomerocytes in the fresh liver cells, they are going to convert it into the metacryptocytes. Just the enlargement of the cryptomerocytes. The size of cryptomerocytes will be enlarged, and they have been called as the metacryptozoids. Okay, now again, this metacryptozoids, they undergo the physogony. Again, the asexual reproduction mechanism. Multiple fission happens here. They undergo the physogony similar to the previous one, producing the enormous number of metacryptomerocytes. Okay, this cycle has been repeated several times, and each time new liver cells are infected. This is how the entire liver cells will be destroyed. Okay, now all these succeeding zygogonic divisions are referred to as the exoerythrocytic or phanerozoic zygogony, and this type of repeating divisions is said to be as the exoerythrocytic phase, and this exoerythrocytic phase is also called as the phanerozoic zygogony. Okay, now this metacryptocytes produced are of two types. The what are all the things? See, initially it was sporocyte, and the sporocyte is converted into this cryptocyte. This cryptocytes undergo zygogony and produce many cryptomerocytes. Now, this cryptomerocytes will undergo a structural enlargement, and that is called as the metacryptocytes. Again, this metacryptocytes undergo the asexual reproduction mechanism called zygogony, and this metacryptomerocytes, they are going to undergo the division called zygogony. They are going to produce two different types of cells. One is smaller and the larger cells. Larger metacryptomerocytes and smaller metacryptomerocytes. What are all the things we are saying like smaller is they are going to give many smaller metacryptomerocytes. Okay, and they are said to be as the micro metacryptomerocytes. They enter the RBC to start the erythrocytic stage. Now, this larger metacryptomerocytes, macro metacryptomerocytes, which is produced less numerous. This smaller will be produced more numerous, and this larger will produce less numerous. And the smaller is called as the micro metacryptomerocytes. This larger is called as the macro metacryptomerocytes. Now, this micro will enter the RBC, and again they are going to start the erythrocytic stage. The next stage of liver zygogony is erythrocytic zygogony. Now, this micro metacryptomerocytes is the one enter into the erythrocytic stage. Next, this macro metacryptomerocyte, they invade the fresh liver cells to continue the exoerythrocytic zygogony. See, this micro metamerocyte, they are going to start the next cycle of the exoerythrocytic phase, that is erythrocytic stage. But this macro metacryptomerocyte is doing again the exoerythrocytic zygogony just to multiply its population. Okay. Next. See, once again, I'm repeating. Initially, it was sporocyte. This is the infective stages. Next, that it is going to convert it into the cryptocyte, and this cryptocyte undergo the process of asexual reproduction mechanism called zygogony, and the end product of zygogony, they are converted into the cryptomerocytes. This cryptomerocytes will enter the fresh liver cells to become the metacryptomerocytes. And now this, what are all the things? The metacryptomerocytes, again they are undergoing many type of they are undergoing the asexual reproduction mechanism called zygogony, and at the end product of metacryptomerocytes, they are giving rise to two different types of cells. One is smaller and another one is larger. This smaller will be more numerous, which means a larger in number, larger in population, and this larger in size, larger metacryptomerocytes will be in the less numerous, lesser in the population. When compared to larger, smaller will be large. And this smaller is called as the micro metacryptomerocytes, and this larger is called the macro metacryptomerocytes. And this micro will enter into the erythrocytic phase, erythrocyte or the erythrocytic cysogony. But this larger or the macro metacryptomerocytes will again going to continue this exoerythrocytic phase. What is the reason? Again, this larger macrometa cryptomerocytes will continue the exoerythrocytic phase, means just to multiply its population. Okay, that's all about this chronological order. That's all about this liver physogony. Clear? And see. Both the phases of the parasite remains immune to the resistance of the host, which means what? This pre-erythrocytic phase and exoerythrocytic phase. Our immune system can't do anything with respect to this multiplication happens in this phase called the pre-erythrocytic phase and the exoerythrocytic phase. They then come to know because there is no type of infection has been seen in this pre-erythrocytic phase as well as the exoerythrocytic phase. As no infection has been recognized by the immune system, it can't the immune system to be the this parasite is the resistant of the host with respect to these two phases. Okay, and as our immune system can't recognize any of the symptoms in these two phases, no parasite, no anti-malarial drugs can be do anything with respect to these parasite only with respect to these two phases. When so the question can be asked, the anti-malarial drugs is very effective in the when the parasite undergoes the process in the liver cysogony, and you have to know when the parasite undergoes the liver cysogony, no anti-malarial drugs and no immune system can do anything against these parasite. Okay, you have to know this. Now, okay, pre-patent and the incubation periods. What is pre-patent and incubation period means? See, this is called the pre-patent period. The duration between the initial sporocyte infection. See, this is the initial sporocyte infection, right? From the infected mosquito enter they inject the parasite or the sporocyte into the normal healthy person. That is the initial sporocyte infection, and the first appearance of parasite in the blood, it is termed as the pre-patent period, which means what? See, initially it is reaching the liver, only the liver. The period from the initial infection and they enter into the blood. This period is called the pre-patent period. Okay, the duration between the initial sporocyte infection and the first appearance of parasites in the blood. Okay, but only when it enters into the human body, the first place they are going to reach is the liver. And the period between the first infection and the parasite we can see in the blood, that period is called the pre-patent period. It varies from species to species. It is about 8 days in the Plasmodium vivax. So the patent period, pre-patent period in the Plasmodium vivax is about 8 days. Now, the period between the infection and the appearance of first malarial symptoms is the incubation period. Which means what? Wait. The period between the infection and the first appearance of malarial symptoms. Now it has been reaches the blood, and after reaching the blood, the person will experience the chillness and shivering and fever. And the symptoms, first symptom, that symptom we humans will experience. That is the period between the infection to the first symptoms experienced by the human. That is said to be as the incubation period. That is also varies about species to species. With respect to this Plasmodium vivax, it is about 10 to 17 days or average 14 days. That's all about the liver cysogony. Now we are going to see the erythrocytic cysogony. And what is the name of the merocytes? Will enter into the erythrocytic cysogony. Just text me. Just chat. We have seen at the end product of the metacryptomerocytes, we can get two different types of cells. One is micro metacryptomerocyte and another one is macro metacryptomerocytes. And only one type of merocytes will enter into the Erythrocyte. Very good. That is micro meta cryptomyrocytes. And this macro meta cryptomyrocytes will again going to continue the exoerythrocytic cycle. Very good. Now, see this micro meta cryptomyrocytes after escaping into the bloodstream. Now, this micro will enter into the bloodstream, invade the erythrocytes or RBC. What are all there are different types of blood cells. We have know that the white blood cells, red blood cells, as well as the blood platelets. But the target area for this parasite is RBC or the erythrocytes, and their erythrocytes they are undergoing the cysogony, that is the process of asexual reproduction. That is why we are naming this as the erythrocytic cysogony. In the erythrocytic cysogony there are different stages have been seen here. The first stage is tropozoid stage and the second stage is signet ring stage. Okay, in the tropozoid stage, inside the RBC, the micro metacryptomyrocytes become rounded, modified into a young tropocyte. Nothing but the structural changes happens here. Now, the micro metacryptomyrocytes enter into the bloodstream and reaches the erythrocytes. There, they are undergoing some of the structural changes. That is rounded and modified into the young tropocyte. And after the stage of micro metacryptomyrocytes, what is the stage we are experiencing is tropozoid stage, where it has been happened inside the RBC. This is what you want to know. The next thing, signet ring stage. As the tropocyte grows in size, now again this tropocyte undergoes the structural modification. It grows in size and the vacuole has been developing here and the nucleus is pushed to one side of the peripheral cytoplasm. These are all the structural changes. Only when this central vacuole has been developed and the nucleus is pushed to the one of the outer side of the cytoplasm, that stage is said to be as the signet ring stage. Why it is called as the signet ring stage? Its structure resembles the signet ring. Okay, that is why this stage is called as the signet ring stage. Now this signet ring tropozoite ingests a larger portion of cytoplasm of RBC. That is why I have explained yesterday. It needs a larger space of the RBC. Sorry, this is not a structure. This is the biconcave structure of the RBC. We know that we human beings doesn't have any nucleus and mitochondria in the RBC. Only then the mitochondria can be multiply very. That is why that is why this parasite will prefer our erythrocytes because of the space and all the contents which is required for this parasite. Everything will be available in the erythrocytes. And why I said in sickle cell anemia patients doesn't they can't have the capacity to experience this malaria. Why? Because the space which is present in the sickle cell RBC will be very low and the mosquito can't undergo any of the further process when it is sickle shaped. When it only the biconcave shaped RBC, the person will experience malaria. Okay, now in that stage, tropozoid stage, in signet ring tropocyte inside ingest a larger portion of the cytoplasm of RBC, forming a food vacuole into which it secretes the digestive enzymes. Once it index, see, you have to remember this porocyte is the infective stage. The signet ring tropocyte stage is the stage where they are going to feed. When it starts to intake food, it needs to digest the food. For that, the central vacuole has been developed. Okay, you have to understand why all these structures will be developed. Whatever be the organism it is, no structure will be developed just like that for no reason. No structure will be developed for no reason. Every structure has their own reason for their development. That is why here the central vacuole will be developed, and that central vacuole is nothing but the food vacuole. Why? Because the signet ring tropocyte stage is the stage where they are going to intake food. And to digest those food, it needs the food vacuole, and in that food vacuole, it has to secrete many type of digestive enzymes, and now this digestive enzyme is going to digest the larger food molecules into the simpler molecules, and after this digestion they have been absorbed. Okay, fine. Besides, the number of pinocytic vesicles are also formed. See, these pinocytic vesicles are also formed additionally. Besides food vacuole, these pinocytic vesicles are also formed around the periphery of the parasite into which digestion has been taking place. Okay, fine. They bring about the proteolysis of blood hemoglobin. They are going to intake what? Hemoglobin, which breaks down. We know that hemoglobin is made up of four different polypeptide chains, and they to break down this, we need a protein enzyme. And now this hemoglobin will breaks down into its protein component and hematin. This protein is used as the food by the trophozoite. Hemoglobin is a protein, is the food for the trophozoite, while the unused hematic forms the toxin material. See, whatever be the thing we have intaking, the main important source will be digested and taken by the and absorbed by the remaining part of the cell bodies. And what are the parts which can't be digested, with the leftover part of the digestion has to be converted in some less toxic form, and it has to be excreted out from the body. But what here is doing, what are all the products taken from the hemoglobin, the useful products taken for its energy, and the unused hematin forms will be converted into the toxic material, and that is called as the hemozoin. What is the toxic material by the parasite is hemozoin. This is the most important one. See, the word sporozoite is the infective agent, infective stage, and the hemozoin is the name of the toxin material. Toxic material. Okay. Now, after this trophozoite stage and signet stage, the another stage we have seen is amoeboid stage. Here, the signet trophozoite stage develops into an active amoeboid stage. When the name comes the amoeboid, definitely what is the organ they are going to develop? Pseudopodia. It sends the pseudopodial process into the cytoplasm of the blood corpuscle. At this time, the small red eosinophilic granules appears in the cytoplasm of the host corpuscle, which are known as the Schaffner's granules. The most important thing. And this is the stage where the diagnosis have been happened. If the Schaffner's granules is present, definitely we are going to identify, definitely this patient will experience the malaria. Okay, see, the signet ring trophozoite will be developed into the amoeboid trophozoite. Now, when the name comes the amoeba, why it is called as the amoeboid trophozoite? And because of the pseudopodia, it pushes the pseudopodia outside of the body to process into the cytoplasm of the blood corpuscle of the host human. At this time, the small eosinophilic granules appears in the cytoplasm of the host corpuscle. This small granules will be seen in the host RBC, and that is called as the Schaffner's granules. Okay, fine. Next, the sysont. This sysont will be developed. The amoeboid trophocyte after active feeding, which is the most active feeding stage, means this amoeboid trophocyte, and becomes a rounder, grows in size and becomes a sysont. Everything is the structural modification. And now again the sysont undergoes many asexual reproduction to multiply its population by the process of cysogony, a type of multiplication. Now its nucleus is divided to form 12 to 24 nuclei, arranged only at the periphery. Everything will be arranged only in the periphery, whatever it is. Okay, and the cytoplasmic masses will be surrounding them. Now, each cytoplasmic masses will be having one nucleus with it. Becomes an oval-shaped merocyte. See, they are considered this as an amoeboid trophocyte. They are undergoing some of the structural modification. They are going to undergoes physogony. This sysont will undergo the physogony, and these are all, each one will be producing the every nucleus, and every nucleus will be surrounded by the cytoplasm like this. Now, this is everything. After this base, everything will be converted into, once again, a merocyte like this. This is a physogony. Now, each cytoplasmic masses with one nucleus becomes oval shaped like this merocyte. And now the hemozoin granules gathered at the center, with the rupture of the RBC, the merocytes will be liberated into the blood plasma. Again, they are going to invade the fresh RBC to repeat the erythrocytic cycle. Now, the ghost. which means what? The leftover part, the left behind the merocytes escape. After it has been escaped, after all these merocytes will be escaped, this remaining part of the thing will be enter into the spleen. What is which organ is said to be as the graveyard of RBC? Just tell me which part of the organ of the human body is said to be as the graveyard of RBC. What is called as the graveyard of RBC? Nobody knows. Spleen is said to be as the graveyard of RBC. We know the lifespan of RBC is 120 days. Lifespan of RBC is 120 days, and after this 120 days, the cell has to destroy. After taking the all important components, remaining parts will be destroyed. And where it is going to destroy? In the spleen. That is why spleen is said to be as the graveyard of RBC. Clear? Okay. Now again, the leftover part of the RBC will go to the spleen, and there they are going to destroy. See, one erythrocytic cycle takes 48 hours in the Plasmodium vivax. It also changes from species to species. That's all about the erythrocytic stage. In erythrocytic stage, another two different. What are all the things seen in the erythrocytic cysogeny means tropocyte stage, signet ring stage, amoeboid stage, and the cyzont. Okay, and after the erythrocytic stage, we have to see this post erythrocytic stage. Post erythrocytic cysogony, which means what? The process which is taking place in the RBC is said to be as erythrocytic cysogony, and after the it has been reaches to the bloodstream, it is going to follow this post erythrocytic cysogony. Again, what is they are going to do? Sometimes some of the merocytes produced in the erythrocytic cysogony, we have seen these are all the merocytes will be formed after the erythrocytic cysogony, and in some merocytes will again going to reach the liver cells and undergo the cysogonic development, structural development in the liver cells, and they again some of the merocytes will going to form the gametocytes. When the impulse to multiply asexually by the cysogony is exhausted, which means what? After the reproduction have been completed, these merocytes do not proceed ahead with the again they are not going to undergo any of the erythrocytic cycle, but after entering the RBC, they are going to convert some of the structural modification like increase in size to become the red gametocytes, and that structure we are saying is the gametocyte. They show the sexual dimorphism, and the gametocytes formed at the end product of the erythrocytic cysogony can be seen as two different type of cells have been seen. One is micro and one is macro. Okay, that is why we are this micro is said to be as the male and it is smaller, and the macro is said to be as the female and it is larger, megagametocyte. As it gives both the type of cells, that is male and female, we are saying the sexual dimorphism. See, the male or microgametocyte is smaller and the female or macrogametocyte is larger. Now, these gametocytes do not divide, okay, because they are ready to get into the body of the mosquito to complete the sexual part of the life cycle. So, the gametocyte do not divide just to wait for the mosquito to take them. Okay, but remain as the intracellular parasites within their host blood corpuscles until they either die or ingested by the vector, that is mosquito, in which they continue their development to continue their sexual cycle. Okay, now asexual reproduction. Now I have explained completely about this human host, what are all the process undergoing within the human host have been completed. Now we are going to see what are all the process will be taken inside this body of the mosquito. Okay, then what is the cycle taking place inside the body of the mosquito means sexual cycle. Okay, now we are going to see the sexual cycle of the Plasmodium vivax of the mosquito, and now the gametocyte will be ready in the bloodstream of the human. Now they are going to enter into the second host body, that is mosquito. Ingestion by the mosquito. When the female Anopheles mosquito bites the infected person, the gametocytes present in the RBC will be enter into the body of the mosquito. The gametocytes, however, they are going to survive. They are liberated and become large in the cavity of the gut. They reach the gut cavity of the mosquito. There they are going to undergo the fertilization. We know the gamete produced at the end product of the merocyte is microgametes and macrogametes. Now, in other animals and most protozoa, reduction division also occurs during the formation of the gametes. Which means what? What is a reduction division? It is also called as the meiosis process. We have studied what is the cycle takes place in the mosquito is sexual cycle. In sexual cycle, always we are going to see the mitosis and meiosis process, and up to the human, we have seen only the mitosis. Sorry, in the sexual cycle, in many of the protozoan, we have seen we are going to see meiosis process. But in this process, all the stages are the diploid. That is, mitosis only happens here, not a reduction division or meiosis will be going to happen. Okay, this only harm gametes are haploid. As male is haploid and female is haploid, once they are getting fused, they are going to give rise to the zygote as a result of fertilization, and the zygote will gain the diploid in condition. Because male has one N and female has one N, and when it both fuse, it gets the 2N. Yeah, but in Plasmodium, this meiosis and reduction division takes place in the first divisions of the zygote. Consequently, only zygote is diploid, while the gametes and all other stages of the cycle is the haploid. What I'm trying to say here is, see, what is the stages? Everything except this zygote and the ookinete stage, it is only this is seen as the diploid condition. All others are haploid. This also can be asked in the question. Okay, you have to make a note of this. Okay, next, microgametes. This microgametes mean the male or microgametocyte begins to undergo process called exflagellation, which means what? They are going to modify some of its structure. In the midgut of the mosquito, they drop in temperature due to the transfer from the warm-blooded human host to the human cold-blooded. The human is the warm-blooded animal and the mosquito is the cold-blooded animal or the poikilotherms. What is warm-blooded animal and the human cold-blooded animal? See, whatever be the external temperature it is, we humans we maintain some of the internal temperature. Always our body temperature will be 36 or 37 degrees Celsius. Whatever be the temperature seen at the external environment, our body has certain own ethics to follow. But in case of mosquito, whatever be the external temperature and whatever be the climatic conditions outside the body of the mosquito, they have to adapt to that particular environment. They do not maintain separate body temperature for their own mechanism. Whatever be the temperature seen at the outside, they are going to adapt to those environment and their internal body temperature will be adjust to the external body temperature. That is why human is said to be as a warm-blooded animal and mosquito is said to be as a cold-blooded animal. And now this parasite has been transferred from the warm-blooded animal to the cold-blooded animal. Definitely, they are going to undergo some of the structural modification to adapt these temperatures. Now, the drop in temperature due to the transfer from warm-blooded human host to the cold-blooded insect provides the stimulation of the process. This shift in temperature, this drop in temperature will provide the stimulation of the process. See, temperature also a factor to affect this reproduction. In each microgamete, nucleus is divided by the mitosis. Again, the division is going to take place and produce 6 to 8 haploid daughter nuclei, which assembles everything. Whatever the thing they are going to produce, they are going to arrange only at the periphery, outer region, periphery. The cytoplasm outgrows into a large, thin flagella-like projections. They are going to produce the flagella, and a daughter nucleus enters each one of them. And these projections breaks away and mature a male or the microgamete. Always the male gamete have the capacity to move from one place to another. Microgamete or the male gamete, they are motile in nature. Macrogamete or the female gamete, they are non-motile in nature. Always the male gamete will reach the female. To reach the female, it has to develop some of the locomotory organ. And this microgamete or male gamete for this parasite, they are going to use the flagella to reach the female. Okay. Now, megagamete are nothing but the female gamete. undergoes some reorganization and becomes a female gamete. Now this is going to ready for the fertilization. Now the fertilization is going to take place by the fusion of the microgamete and the female gamete. Now what is going to produce? This zygote will going to produce. The megagamete gives out the small cytoplasmic production, the cone of the reception of the fertilization cone, which means what? The male is, this is considered as the female, and this is male. The male is going to reach the flagell, reach the female. To reach the female, it has some locomotory organelle or whatever it is going to reach the female. And the female has to receive that. To receive the male gamete, it has a some of the structure called the cone of reception, and that is also called as a fertilization cone. Now, the nucleus of the female gamete comes to lie near its receptive cone, and near this fertilization cone, the nucleus of the female gamete will be present. When the lashing microgamete comes into contact with this megagamete, when the male gamete come to contact with the female gamete, they are going to receive the nucleus of the microgamete. Now the complete fusion of the nuclei will occur as well as the cytoplasm of these two gametes will occur, resulting in the formation of the zygote. Okay, now this syngamy is, I have explained when I read the when I studied about the protoplasmic reproduction. In that we have studied about the syngamy. In that syngamy, I'm explained different types of based on the types of gamete. I have explained the different name, anisogamous, isogamous, like that. In that anisogamous, what is anisogamous? Is male is small and the female is large. Because the structural morphologically it is different, it is called as the anisogamous. Now, ookinete. The zygote remains rounded, motionless for some time, but soon it becomes the elongated, vermiform, and the motile. It performs a wriggling or the gliding movement and is known as the vermicule or the ookinete. Now, after the zygote has been formed as a result of fertilization, the ookinete will be going to form. Now the zygote will remains, the zygote is rounded, motionless. Now the zygote becomes round in shape and motionless. It can't move from one place to another for some time, only for some time. But soon they are become elongated, vermiform and motile. Which means what? Initially the this male gamete is motile, this female is non-motile. After the fusion of the cytoplasm, the nucleus and cytoplasm have been fused and this zygote will be motionless. This is called a zygote. And after this zygote, which is motionless, it gains the motile capacity and it is called as the ookinete. And this performs a wriggling. 1 minute. Sorry, because of the external sound. Okay. Now, this zygote will gains its capacity to move from one place to another. That structure, it performs the wriggling or the gliding movement. This is called as the vermicule or the ookinete. Now, the structure is not important and this is also not that much important. Now, this ookinete is the next stage of kinete which gains the capacity of the motility. Okay. Now, enchestment. Now, this ookinete penetrates through the wall of the midgut. This is the end, but this ookinete is the end product of the parasite which is present in the midgut to settle down just under the thin membrane that separates the midgut from the hemocyte. Here it becomes the spherical and begins to encysted. Again, they are going to undergo some of the structural change. What has been done in the parasite? These ookinete have been covered by the cyst wall. That is the process called the enchestment. During the favorable condition, then this encystment will undergo the process of excisement, and they are going to reach the salivary from the salivary glands to the normal healthy person. Now, this cyst wall is thin, membranous, and elastic, secreted partly by the ookinete and partly derived from the midgut tissues of the mosquito. The encysted zygote is called as the oocyst or the sporozoon. Okay, after the fertilization, zygote will be formed. After the zygote, ookinete will be formed. After the ookinete, encystment process of encystment will be happened, and this encysted zygote will be consist as the oocyst or the sporozoan. You have to well aware of the terminology. The terminologies can also be asked in the question. The oocyst grown in size, now they can now be seen on the outside of the midgut or the stomach. They are transparent in nature. Okay, they may be 450 or more in number. Now this porogony. Again, they are going to undergo the asexual multiplication mechanism called the sporogony. Nucleus first going to devise the meiosis process, that is post-zygotic, and after that mitosis will be happen. This can also be asked everywhere. First thing, mitosis will be taking place. After the mitosis, meiosis will be taking place. But here in this malarial parasite, what they are doing, initially meiosis will be happening and followed by the mitosis. At the same time, the cytoplasm develops a large vacuoles and taken up the sponge-like structures in which the number of irregular cytoplasmic masses are formed connected by the protoplasmic structure. This is not that much important. Just know they are undergoing again the sexual multiplication. First, meiosis will be happening, followed by the mitosis. And this meiosis is also called as the reduction division mechanism. Now, this each sporocyte has been tapering ends. This is the structural modification of the sporocyte, and the sporocyte is an infective agent. This has been already seen in the previous slide. You had referred to that. Okay, the same thing will be repeated there. And human malaria, we have studied 4 different species are going to cause this, responsible for this malaria. Of all the contagious form caused by the protozoan, malarial diseases are the most destructive for the human. We have seen the 4 different types of malaria caused by different species of the malaria protozoan parasite, that is tertian, benign tertian, and the vivax malaria. See the other names. The other names are also important. They can be asked in the match the following questions. Now, the pathogen of this, what is the causative agent for tertian, benign tertian, or the vivax malaria is Plasmodium vivax. And their characteristic feature is fever for every third day. That is after 48 hours. This after 48, before this, up to this 48 hours, we can call this as a pre-patent period. And after the 48 hours, the first symptom have been experienced by the human body. So we can call this as an incubation period. Of course, this vivax malaria caused by the Plasmodium vivax is not fatal. It has been seen only in the temperate regions. Next comes to the quartan malaria. This is the causative agent called Plasmodium malaria is the causative agent for the quartan malaria. And what is the characteristic feature is fever for every 4th day after the intervals of 72 hours. We know that pre-patent period and the incubation period will be vary from the one species to another species. Of course, this is not fatal, but it gives rise to kidney related issues. Clear? And this is confined only to the tropical and the subtropical region. See, this quartan malaria has the capacity of relapsing. What is relapsing? It is well known for its longevity. The unique feature of a quartan malaria is if it is untreated, again the disease have been formed. That is, again the disease will gain its favorable conditions, and even after 40 years the person can again experience this type of quartan malaria, which is caused by the Plasmodium malaria. Now, ovale or the mild tertian malaria, this type of malaria resembles very much to the tertian malaria caused by the Plasmodium ovale. See, name of the disease and the causative agent is important. The characteristic feature is recurrent fever for every 3rd day at the interval of 48 hours. It is not harmful. Okay, mainly confined to the tropical Africa. And estivo-autumnal malignant tertian or pernicious malaria. This is the causative agent of this malaria is Plasmodium falciparum, and this falciparum is the only thing it is considered as a fatal because it's affecting the brain. Once it affects the brain, no other functions have been processed by the human, so of course it will be causing death. And the characteristic features of this Plasmodium is fever for 48 hours, after 48 hours. Now, Quotidian malariae, when more than one species of Plasmodium infected the patient or when two or more generations of the parasite mature on successive days, the fever is repeated daily with an interval of 24 hours. Even within after 24 hours, we can experience a fever when multiple species of Plasmodium will affecting the human body, and that is said to be as the Quotidian malariae. Clear? And symptoms, we know that cold stage, hot stage will be there, chillness and shivering of fever will be there, and fever degrees high, how sudden drop in temperature will be there, sudden higher in temperature will be there. See, this cold stage will last for 20 minutes to 1 hour. This higher in temperature, the person will experience sudden chillness and shivering, and the person after 1 hour, the person will again experience a severe fever, that is 41 degrees Celsius or 106 Fahrenheit. And this stages will experience 1 to 4 hours. What I'm trying to say is 20 minutes to 1 hour, their experience is chillness, they're shivering. And after the 1 hour, 1 hour to 4 hours, they will experience this rise in body temperature. And after this hot state, they experience the sweating stage. As the temperature lowers down, the patient sweats profusely. So sweating is also another symptom. This will also experience 6 to 10 hours depending upon the species of the plasmodium. Okay, fine. And this, we know that what is the toxin material? What is the name of the toxin material of this malarial parasite? Just text me quick. We have to solve the questions. Anjanelu, Lalita, just tell me. Very good. The name of the toxin of the malarial parasite is hemozoin. Okay. And if you read this PPT, you can experience an easy thing. Okay. Everything I have explained before. Okay. How we can has to be controlled means elimination and destruction of the vector. How we can destruction the adult mosquitoes means by killing by hands, which means what is the traditional method? What we'll do? Generally, when mosquito will bite, we'll beat the mosquito. Okay, and before we beating it, fly away. Okay, that is gentle method, killing by hands and traps, fumigation, and you have to read what are all the fumigation agents we are using. Now, that after fumigation, spraying by some of the chemicals, and this is the method followed by our government. Okay, and sterilization method. This usually practiced in the Japan. This is very effective actually in controlling mosquito population. This is very effective. And after we can, by destructing the adult mosquitoes, we can eliminate malaria and eliminate the breeding places, especially the marshy areas. See, generally when the dengue season will come, the government will do what wherever the stagnant water will there, they'll they will spread the bleaching powder into it just to avoid the malaria or the dengue. Okay, and so the watery areas will be the breeding places for this mosquito. Okay, now we can also destroy the larva and the pupa stage of the mosquito by maintaining the proper drainage, by preventing this oil screens. Okay, generally oil spills will be formed in the watery areas. So these type of these things has to be prevented. By preventing this, we can destruct the larva and the pupa of the mosquito. Now, chemicals also have been used. You have to read the chemicals. What are all the chemicals they have been used? This can also be asked in the question. DDT, DDD, benzene hexachloride are very effective in the larvicides, and Paris green, DDT, BHC effective in control the surface feeding place of the Anopheles larvae. Now, the extract of aquatic weed. Ceratophyllum dermersum. This is the aquatic weed. From this, we can control the malarial larva. See, tulasi, Ocimum sanctum, seeds, they are also introduced in the water to control the mosquito larva. These are all the chemical methods. And what are all the biological methods? Generally, according to me, biological methods is harmless, because what are all the insects, what are all the things, what are all the organisms which are going to eat the mosquito? If we introduce those things, Where the mosquito population will be high, the mosquito will be eliminated easily without harming any others, right? So biological methods will also always be safe, and your work is what are all the things which are natural enemies, and who is going to eat the mosquito. You have to aware of those organisms, and how we can prevent the infection um, by some hygienic methods. You defense against mosquito bites, building house on high grounds. If we a certain if the if you're in the upstairs means the mosquito population will be very low. If you are in the ground floor means the mosquito population will be very high, so the mosquito can't fly to some of the heights. Okay, that is why building house on high grounds, we can we can avoid avoid mosquito bite. Mosquito net, the cream, some of the repellents, these are also have been used. And prophylactic drugs, what are all the drugs? Quinine, paldrum, daraprim. Yeah, this I have been explained yesterday itself. And you have to read what are all the drugs and what are all the how can we reduce the susceptibility to the infection by avoiding exposure to the bad conditions, by avoiding whenever we are going to see where the mosquito population will be high, we have to take some precautions, we have to take some safe equipments with us, and we have to reach that place. Okay, how we can read the quinone? Quinone is the main. These are there are the list of drugs, and you have to read those list of drugs. Anything has been struck in the question. Okay, and mainly before a long time, quinine is quinine, which is extracted. Quinine is the name of the drug which has been extracted from the bark of the cinchona tree, and this cinchona tree will be grown only in Java, Peru, and Ceylon as well as in India. Okay, but after the Third World War, the Japan got control over these areas, and no other countries will be entered into those areas. So we have to make an alternative for this quinine. So scarcity of this quinine will lead to the production of other synthetic drugs like quinine, atabrine, camoquine, chloroquine, mebaquine, like this. Okay, you have to read those name of drugs. And the most potent drug is daraprim. Okay, it is a pyrimidine and the single dose of the 25 mg. You have to know. Okay, fine. And government has some of the anti-malarial campaign also. This is not that much important. What I have mentioned here is what are all the research institute will continue their research in elaborating the in eliminating the malaria. Okay, that's all about the malaria. Malaria has been completed. Oh, any doubts? You can ask me if there is any doubts. Anjaneyulu, any doubts? Lalita, doubts? Okay, if you have no doubt means let us. Okay. Uh, shall we move on to the questions? See, in your exam, in the last DL exam, you have get some questions. Okay. See, answer this question. Choose the incorrect statement from the following. I have completed in the general concepts. Let me read the question. Choose the incorrect statement from the following statement with respect to the coelom in animals. They are asking the question about the coelom. What is coelom and their incorrect statements. Only one statement is correct in this here. Coelom separates body wall from the gut wall. Coelom is a extensive internal space. Coelom is developed in diploblastic animals. Coelom provides hydrostatic skeleton in all coelomates. What is the answer? What about Anjaneyulu? Okay. See, what is coelom? Just tell me the exact word of the what is meant by coelom. I have explained you when I explained the syllabus as well as I have explained in the general concepts. What is coelom? Body cavity. Okay. You are saying body cavity in the is the rise. What is body cavity? What is cavity? They are the internal space. Okay, body cavity, and they are going to develop where there is a diploblastic animal. See, in the coelom, we have said two different types of coelom is there. Two different types of coelomates animals are there. One is isocoelomates and another one is enterocoelomates. What is this isocoelomates? The body cavity will be arise by splitting the mesoderm. This enterocoelomates means the body cavity will be arise when by from the digestive tract. So obviously they are going to develop from the diploblastic animal. When it possesses the uh, triploblastic animal, already there is an ectoderm, endoderm and mesoderm where the body cavity will going to arise. Okay, so they are developed in the diploblastic animal. Definitely cavity is nothing but internal space. Okay, and they are going to separate the body wall and the gut wall. As I said, enterocoelomates, they are going to separate the body wall and the gut wall. All the three options are right, and the wrong statement is, do I explain anything about the skeleton? Coelom provides hydrostatic skeleton in the coelomates. As it separates the body wall and the gut wall, it is going to present in the middle area. How it can be provides the hydrostatic skeleton? So this will be the wrong answer. Clear? Any doubts? Any doubts? Wait, we are going to do next question. Okay. Now, okay. Answer for this question, the causative agent of the infection in human due to the contamination of drinking water with sewage. Lalita, Anjaneyelu, answer for this question. What about another one? The causative agent of infection in humans due to contamination of drinking water with sewage. Look at this word sewage. Anjaneyelu, answer. Okay. See, Plasmodium vivax, we know that how these are going to transfer. Everything will be transferred due to the unhygienic condition. But due to the contamination of drinking water with sewage is Entamoeba histolytica. Because of this, we are getting the amoebic dysentery. If we are getting contaminated with any sewage condition, means they definitely our body will eliminate from our system, and that is said to be as the dysentery. And that amoebic dysentery will be caused by Entamoeba histolytica. Clear? Okay. See, the we have completed protozoa. We have completed protozoa, and the how the questions have been arising here. So we can easily scrutinize the options only when you repeatedly reading the PPT. Okay, so don't avoid the PPT. These are all the two questions in the last year I have sent. Another question. There are many questions from core data and core data. In that only in one line we have getting two different in general concepts and in the only one line of protozoa in the syllabus we can get two questions. So see the importance of the topic. So this is how you have to read. Now You can answer this question. Sleeping sickness is caused by. Why are not? Why are you? Okay, okay, fine, very good. The answer. What is the species name of trypanosoma? Tell me the species name. It is Trypanosoma gambians. Trypanosoma gambians is the one causing the sleeping sickness. Okay, fine. Yes, very good. And Entamoeba histolytica is going to cause the what is it? Entamoeba histolytica is going to cause the amoebic dysentery or the amoebiasis. Plasmodium is the one going to cause or the malaria. Leishmania is the one going to cause the American sleeping sickness. Okay, this is African sleeping sickness. And polymorphism is shown by polymorphism is shown by in which I will give the hint in which a parasite we have said the different forms this parasite can exhibit in different forms. Do we see anything cycon like that? We didn't. I didn't even teach porifera. Okay, this is the cycon the canon system of porifera. Cycon is the cycon type of sponges. Okay, just you tell me the question what I have been explained. Okay, there is amoeba is there, paramecium is there, trypanosoma is there. When we are discussing the protozoan questions and you are giving the answer as icon. No problem, just relax and answer this question. In which parasite I have explained the different forms. Is it paramecium? In paramecium, do we see anything as the amastigote condition, critidial form, trypanozoid form? In which thing we have seen these type of forms? Lalita, answer. I have explained this question many times, but you are not answering. See, polymorphic forms of the trypanosomes. Leishmanial, amastigote, leptomonad, critidial, trypanosome. These are the different forms of the trypanosoma, right? Don't get confused, okay? This is the simple question. Okay, very good. Okay, if you read the PPT means you can easily answer the question. See, everything will be there in the PPT. Okay, trypanosoma is your answer. Slipper animal give. This is also I have explained. It's look this and this organism, this protozoan looks like a slipper, like this. And what is it? Lalita answer. Very good. Paramecium. Clear? And now, paramecium chordatum has everything. This also I have explained. Paramecium chordatum has a single nucleus, two micronuclei of the same size, one micronucleus and one macronucleus, only one meganucleus. See, this protozoan has two different types of nucleus. One is micronucleus and macronucleus, and one two micronuclei of the same size. Did I explain anything like that? See, there are paramecium is the one has the micronuclei and the macronuclei. See, macronuclei in the paramecium, we have two different types of nucleus. Okay, one is micro, smaller in size, macro, larger in size. Okay, this micro is responsible for the reproduction function. I have explained when I teach you the conjugation process. This is in the macro, it is responsible for the vegetative function. Okay, in paramecium, we have two different types of nucleus. One is micro, one is macro. One is small and one is large. The small micro nucleus will be responsible for the reproduction and the large macro nucleus will be responsible for the vegetative function. So, the paramecium chordatum has the one micronucleus and the one macronucleus. Clear? Fine. The paramecium and other ciliates differ from all other protozoans in having two types of nuclei, capturing the prey with pseudopodia, having contractile vacuoles, not using flagella for locomotion. Just now we have seen. Just now we have seen. If you know the last question, if you understand this last question completely, you can answer this question easily. How the paramecium, we know there are different phylum, subphylum seen under the protozoa. There are Sacromastigophora, Sporozoa in Protozoa, Sacromastigophora, Sporozoa, Mediophora, and Ciliophora. In that Ciliophora, we have a paramecium and other ciliate examples are there. The questions comes from here. How this paramecium will be differ from other ciliates. How it is making the difference means this only paramecium have two types of nuclei. Okay, only this paramecium has the two type of nuclei. So the answer is having two types of nuclei. Clear? Fine. Which of the following statements is not true of the paramecium? They are the predatory ciliates. They use pseudopodia for capturing the prey. They are preyed upon by didinium. They use contractile vacuole for removing excess water. Not true about the paramecium. You can easily guess as it is comes from the ciliophora. What is their locomotory organelle? Definitely they are going to use the cilia for its locomotion. Okay, see, this is why how the classification, how the understanding about the classification helps in answering the question. This is why I am stressing. If you know the outline of each and every word of the syllabus, and if you know that at least 5 to 10 important characteristic features of each word in the syllabus, you can easily crack the exam. Okay? Not true about the paramecium means as it comes from the ciliophora, it is definitely going to use the cilia for its locomotion. So obviously this statement is strong and that is the correct answer for this question because the question asked here is not true. Now, animal-like nutrition is known as holophytic. If you know the name, if you know the meaning of each terminologies, you can answer this question. Okay, what about the other one? Anjnelu answer. Very good. Holozoic nutrition. If they are plant-like nutrition, they are holophytic. If they are animal-like means holozoic. If they are host-like means it is parasitic. If they are dead and decaying organic matter-like means saprophytic. Clear? Fine. And the answer for this question is holozoic. How paramecium takes its food? How paramecium takes its food? By pseudopodia, by oral groove, by undulating membrane through body surface. I'm sorry. How paramecium takes its food? See, definitely pseudopodia can't be the answer. Okay, body surface, undulating membrane. Because if the flagella is present, we can guess the undulating membrane because of the undulation motion. Protozoa has the protozoa comes under the protist. Protist has the unicellular eukaryotic in nature. It has a well developed organelle in it and it has the oral groove structure. By the oral groove, it takes its food. Clear? The answer for this question is option A. And cyclosis is present in. Cyclosis is present in. Cyclosis again, it is present in the paramecium. Okay, see, based on the syllabus, we can cover the syllabus, but these are all the questions. Why I have included in these type of question means, anyhow, solving these questions will help you in your examination to eliminate the option. Okay, you by solving this question, you know this paramecium has take its food by the oral group. By you can get some idea when the difficult question asked in the exam, at least you can eliminate the option. That is why I'm explaining what are all the things which are not in the syllabus. Of course it is comes under the paramecium, of course it is comes under the protozoa. You have to know all the important protozoans, example euglena and amoeba. If you know about the protozoa, the only examples every other aspirants will preparing for the exam read will be only amoeba, but you should know some of the important things about amoeba, paramecium, euglena, and some of the important protozoans. Okay, for example, in paramecium you have to know the oral groove will be present for their food. For amoeba you have to know pseudopodia will be helpful, like that. Okay, and like in the same way, cyclosis is present in the paramecium. Okay, paramecium is the correct answer. And the okay, you take this word and you put as the protozoa. The food vacuole of protozoa is, I have explained you this, to digest the food. Half of the digestion will be takes place in one of the pH, and remaining half of the pH digestion will be takes place in the another pH. And what will be the pH? How it is acidic or alkaline or both? What is it? Uh, by seeing this, I can easily know nobody reading the PPT. Yes, first acidic, then alkaline. Okay, the proteins in the acidic medium, it will be converted into the dipeptide. Okay, now from this dipeptide will be converted into the simpler amino acids in the alkaline medium. Half of this digestion will be takes place in the acidic medium, and this half will be takes place in the alkaline medium. Okay, you have to remember this. First acidic, then alkaline. Clear? Now, intracellular digestion occurs inside. Intracellular digestion occurs inside. Very good. Yo, I don't mean to hurt you or something like that. I don't want to hurt you, but you have to, after this is the procedure, what I'm conducting the classes, it is the perfect way to clear any of the examination. Not only this, any of the competitive examination. You have to read. For example, if we read the protozoan general characters and characteristics, you have to solve some questions. After locomotion, I have solved some questions. After the nutrition and reproduction, I've solved some questions. After this disease, after each disease, I've solved the questions. And after completing all these, I'm conducting of the test. And what is your duty? You have to make use of it, and you have to read the PPT repeatedly. And if I am taking the class very slowly, this is slow, only slow. Okay, it is not fast. So you can easily cope up with me, and you have to complete the syllabus. When I teach the complete one topic, you have to read those in the PPT immediately on that particular day itself. It will be useful at the end of the day. Okay, and intracellular digestion will be occurs inside food vacuole. Very good. Okay, next. What is the main function of the contractile vacuole? We'll know that. Option A, food regulation, extraction, osmoregulation, and reproduction. Main function of contractile vacuole, it is obviously osmoregulation. Asexual reproduction of paramecium is brought about by paramecium. Is the paramecium. This is the cut. This is this upper part will give rise to one paramecium, and this lower part will give rise to one more this this another paramecium, and this cut is called as the transverse. If I cut a paramecium like this, means longitudinal. Okay, and this can't be happen, only this is going to happen, that is called as the transverse medium. As this one paramecium gives only two types of two daughter paramecium, so it is called as the binary fission. So multiple fission is not the answer. Sporulation is not even possible. Simple binary fission is not possible because it has to cut at the transversely. So transverse binary fission will be possible. Okay. Next, which of the following protozoan lives in the gut of the termites and helps them to digest cellulose? It is trichonema. I didn't explain this question. I didn't explain in when I'm teaching. I didn't explain this, but you have to know this. Okay, if you didn't know, there is no problem. Just aware of it. Okay, when the following protozoans lives in the gut of the termites and helps them to digest cellulose. So what you want to know is when I teach about the protozoan nutrition, protozoa has the capacity to digest the carbohydrates. It has the capacity to digest it has the capacity to digest proteins, and it has also the capacity to digest the fats. You also know the protozoa also has the capacity to digest the cellulose. Okay, so what you want to know is which of the protozoan, this protozoan trichonympha, lives in the gut of the termites. Okay, where it is going to live in the gut of the termites and helps them to digest the cellulose. So trichonympha is the correct answer. These are all the unique features. So you have to know these type of protozoans also. Glossina palpalis transmits the infective stage of Glossina palpalis is the tsetse fly. Biological name of the tsetse fly is the Glossina palpalis. They helps them to transmit the infective stage of tsetse fly is the carrier of which type of disease? Very good. Trypanosoma. Trypanosoma gambians. Clear? Fine. Next. Chagas disease is caused by. Chagas disease is caused by. I didn't explain you this. Just know this Chagas disease is caused by the Trypanosoma cruzi. Okay? Fine. Fine. Next. Infective stage of Entamoeba histolytica is. Infective stage of Entamoeba histolytica is. Okay, don't confuse between the infective stage of Entamoeba histolytica and infective stage of malarial parasite, Plasmodium. See, infective stage of Entamoeba histolytica is sporocyte. Sorry, tropocyte. Sporocyte is the infective stage of the protozoan disease called the malaria. Okay, now only I have explained malaria. Sporocyte is the infective state that have been transmitted from the salivary glands of mosquito to the liver cells of the human, and that will be the Sporocyte. And what the question have been asked here? Infected stage of entamoeba histolytica. So the answer will be the tropocyte. So you have to revise the protozoan diseases right from the beginning. Yeah. How does the locomotion takes place in the entamoeba? The locomotory organelle takes place in the entamoeba histolytica is just what is the locomotory organelle? Just any guesses? Very good. Lobopodia. Very good. Next, the disease Kala Azar is caused by Trypanosoma, Giardia, Trichomonas, and Leishmania. You have to know this disease Kala Azar is caused by the Leishmania donovani. Very good. Leishmania donovani. Clear? Fine. Next, the adult feeding stage in the life cycle of monocystis. This is not E, this is C. The adult feeding stage in the life cycle of monocystis. Monocystis is also the sporozoan category. Sporozoa of the protozoan, and the feeding stage in that life cycle is. I didn't explain this. So the active feeding stage of this life cycle of monocystis is sporocyst. Okay, as the name suggests, if you know this monocystis will be comes under the sporozoa. I have explained about this class. The sporozoan, which the protozoan, which uses spores as their better asexual reproduction mechanism. If you know this monocystis, I have explained you to read the protozoan classification and its examples. And this monocyst is comes under the sporozoan, and I have explained what is the mechanism, spores. And the spores is the perfect asexual mechanism for these type of protozoans. If you know this basic understanding, you can easily answer this question. Okay, fine. Amoeba has which type of pseudopodia? These are all the types of pseudopodia. And amoeba has which types of pseudopodia? Once again, lobopodia is the correct answer. Sleeping sickness is caused by sleeping sickness is caused by tsetse fly. Okay, sleeping sickness, very good. Sickness is caused by the tsetse fly. And next, in paramecium, the nutrition is the paramecium, the nutritionist, paramecium also prefers to each another organism. So obviously this paramecium nutrition is holozoic. Okay, we know this paramecium chordatum has two types of nucleus, one macro and one micro. Okay, one macronucleus and one micronucleus, and the only protozoan which has a dimorphic in nature. See, another word for these two different types of nucleus is dimorphic. This also you have to know. If you read only this one macro, one micro, and you forget this technical term, means there is no use. Okay. So the organism which possesses these two different types of nucleus for their entire life cycle means it is dimorphic. One macro and one micro. And macro is responsible for the vegetative structures, vegetative functions. And this micronucleus is the one responsible for the reproduction function. Okay, fine. African sleeping sickness is caused by, again, the same question is repeating. African sleeping sickness is caused by the trypanosoma, and it causes amoebiasis. Obviously, entamoeba histolytica is the one going to cause the amoebiasis. Cysogony and the cysont stage of plasmodium occurs in. Answer for this 52nd question. In today's class, I have explained. That is why I have keep on stressing you to read the end product of each organ. Cysogony and cysont of plasmodium occurs in. See, you have saying the blood of human beings. In blood, what specifically? Which cell specifically? RBC, and that RBC is said to be as the erythrocytes and liver cells. So obviously the answer will be erythrocyte and liver cells of the human beings. Okay, cysogony and cysont stage of plasmodium occurs in erythrocytes and liver cells of the human beings. Even if you don't know the any of the process like this, if you know what is the target area of the human beings, liver cells and erythrocytes, if you know that basic understanding, you can easily answer this question. Okay, fine. Exflagellation in malarial parasite takes place during the formation of exflagellation. Okay, exflagellation in the malarial parasite takes place during the formation of. The answer for this question is sporocytes. And your duty is why the answer is sporocyte. You have to read the PPT and you have to explain me tomorrow. Clear? Fine. And answer for this question. Sporogony and the malarial parasite occurs in. You have to find the answer for this question. This will be the homework for you. You have to answer for this 54th question and you have to explain me why the sporocyte is answer for 53rd question. This two will be the homework. I'll upload the PPT in the group and you can make use of it, and protozoa has been completed, and if you read the PPT by today itself, it will be easy for you at the end of the day. Okay, fine. Any doubts means you can ask now. If there is no doubt, you can leave the class. Anshnelu, is it clear for you? And if you want to make some changes regarding the class, how the classes has to go, if my methodology is okay for you means, you can say yes, or if you need anything to improve means you can say me now. See, please be quick. Okay. If you have no doubts, no, yeah, I can understand. Okay. If you can't attend the regular class, at least go through the recording class. Sir will upload the recording in the. Don't worry. Just attend the class and just listen the class. Everything it is perfect. Just listen the class, and that 1.5 hours will be make complete focus in the class. That is more than sufficient. Okay, and the class. Okay, sir will upload the recording in the app. Okay, fine. Thank you. Lalita, are you clear? If you have any doubt means you can post me now. If not, we'll wind up the class. Okay, fine. Thank you. Revise the PPT once, and tomorrow sharp 6 a.m. we'll meet in the class.